Welcome to webinar 7 of Platomics IVD Ready program, which we have given the title, The Real Risk is Not Managing Risk. My name is Simone Latkulik. I'm leading the customer success team for labs at Platomics, and I will guide you through today's webinar. The difficulty of risk management under IVDR is a challenge for many diagnostic labs. Risk management is not only about setting up a risk management file for each in-house test, it is also about establishing a process for updating it over the whole life cycle of the test. This webinar provides orientation. We will outline IVDR requirements for risk management processes and documentation so your lab understands what it needs to do. Let's have a look at the agenda on our speakers. Today, we have invited the following speakers. Andreas Oberleitner, Chief Regulatory Officer at Platomics, will introduce the topic risk management and its regulatory environment and framework. Later, he will show the latest PlatoX IVD Assistant module for creating a risk management file. Andreas will perform a demonstration for us of the PlatoX Risk Management Studio that will be released in May on our PlatoX platform. I'm very pleased to announce our guest speaker, Professor Florent van Stapel. Professor van Stapel is a member of the European Federation of Clinical Chemistry and Laboratory Medicine, where he serves on the Task Force for European Regulatory Affairs, as well as the Working Group, Accreditation and ISO Standards. Professor van Stapel studied and trained at the University of Leuven in Belgium. He was active in general clinical chemistry, inborn metabolic disorders, and point-of-care testing. He will talk about IVDR-compliant risk management for lab-developed tests in practice. On the agenda of this webinar is also a QA dialogue, a session with Professor van Stapel. You can ask questions in the Q&A section during the webinar if you want anonymously, and we will try to answer them by the end of the webinar if we have enough time. We are looking forward to an interesting discussion. Today's webinar is part of our IVD Ready webinar series, which takes place in parallel to the progressive rollout of the IVDR. If you are interested in our past webinars, you can find the link on our homepage and can watch them on demand. Finally, we will have our last webinar of the IVD Ready series on June 4th, entitled IVD Ready to Go. Here, we will offer an outlook on important aspects for remaining IVDR compliant, including an up-to-date account of the final phase of implementation and what it means for labs. If you want to get PlatoX access, you can register for the PlatoX IVD Assistant by typing in the chat window below or by sending an email to us at info@platomics.com. Now, let's start with an introduction to the topic by Andreas Oberleitner. Andreas, now over to you.
Thank you, Simone. Good afternoon from my side from Vienna. Before we start and enter the topic, I would like to give you a very brief update or an overview on the regulatory landscape, especially with regards to the risk management. So from the point of view of laboratories creating their in-house devices. First of all, we know that in the core of the regulation, we have the IVDR, the regulation, which also states requirements for laboratories as being in-house manufacturers. So as we all know, in-house devices are now regulated under Article 5.5. So first of all, we need to consider that and analyze this article. Furthermore, this article immediately links us to the Annex 1, the General Safety and Performance Requirements, and these General and Safety Performance Requirements already contain a lot of different requirements representing the risk topic. Then we should not forget about potential national laws, which could also add some more requirements on top potentially. And since the beginning of last year, we have an MDCG guidance document in place, which helps us also in the interpretation of several aspects of the IVDR. Regarding the standards, which represent the state of the art, first of all, I would like to mention the ISO 15189, the standards on the requirements for quality and competence, which is, by the way, also directly mentioned within the IVDR Article 5.5. Furthermore, there is a related standard, so ISO 22367. This standard has the title Application of Risk Management to the Medical Laboratories. So we see already right now it's a very specific one, and those two standards fit pretty well together because they have been developed in the past also together. But there is also another standard coming from the more medical device domain, the ISO 14971. This is mainly focusing on manufacturers of medical devices. However, the risk concepts are pretty similar, and I also would like to talk about this standard a little bit today. Last but not least, we see that there is another draft standard already in the making, which is the ISO 5649. This will become a very specific ISO standard, a global standard on the concepts and specifications for design, development, implementation, and also the use of laboratory developed tests, LDTs. I just did a quick review, and as you can see here regarding these figures, the appearances of the word risk, we see the topic is really pretty important throughout all of these regulations and standards. So the word risk appears 230 times in the IVDR, only 62 times in the Annex 1 alone, so pretty important already. Of course, we also see that the relevant standards dealing with risk, of course, have also a high appearance, but even ISO 15119 is already mentioning the word pretty often. Before we enter into the topic, I would like to discuss some basic terms. First of all, what is a risk? What is a hazard? Before we start talking about a risk, we need to understand what the hazard itself is. So the hazard, by definition, is a source of potential harm. Nothing yet happened. Like the shark, if it's not close to you, then it might not harm you directly. For this, it needs a hazardous situation. So these situations, by definition, lead to a circumstance in which someone can be exposed to one or more hazards. For example, if you're going to swim or dive with the shark. At the end, this hazardous situation may lead to harm, which is the direct damage to physical damage, to what you can suffer physically, but also psychologically at the end. On the other hand, how to measure all of these things? On the one hand, we are talking about severity, which measures the possible consequences of a hazard. The probability is the chance of occurrence from nearly impossible or impossible to absolutely certain. Something in between, in most cases, is the truth. And this combination at the end makes the definition of what a risk is. So the risk at the end, from the understanding of the regulation, is the combination of the probability of occurrence of harm and the severity of that harm. The higher each value gets, the higher the risk will be. These risks, from the point of view of the regulation, need to be minimized to a certain level. We will see that later, and this is called the risk mitigation. But nothing in this world will come without any risk. So that means there will always be residual risks which remain, and you need to balance and also to match against the potential benefit of your systems. So then let's jump directly into the requirements, starting with the basic requirements, again, from our famous Article 5.5. We know that on the one hand, it's directly referencing to Annex 1. So that means we need to have a look into Annex 1 and also fulfill the applicable requirements, first of all. Then we see that there is another point pointing to one of the standards I already mentioned, the EN ISO 15189. So the next requirement definitely will be to be compliant with this standard, or where applicable, national provisions, which might be pretty similar. Then we also need to draw up a declaration. This declaration should state that the laboratory fulfills all of the GSPRs, all of these requirements. So this is a more or less formal and also official and publicly available confirmation that Annex 1 is going to be met. Last but not least, the health institution also needs to review the experience gained from clinical use over and over again. So therefore, what you need to implement, of course, it's a living process. It does not stop until a certain decommission of the product would happen. Let's have a look into the Annex 1 itself. So especially the first chapter is dealing a lot with the topic of risk management. The first requirement, number 1, is already one of the broad and very general ones, but pretty important ones. So it's at the end, you do not need to read through all of these texts, just concentrate on the red frames. But at the end, it asks you to do a certain risk versus benefit analysis on your product. Additionally, it wants you to reduce the risks as far as possible. This AFAP model here that we see, this is really important to understand. So there are certain circumstances where you even need to reduce the risks down to a level, and you cannot bring any economical reasons for not doing any further reduction, theoretically. So that means that on the one hand, you have to balance it against the benefit. On the other hand, you need to demonstrate that you have done everything which is relevant to bring it down to a level as far as possible. You also need to consider new risks which can be derived from putting in mitigation measures as well. You need to set up a whole risk management system. So this consists, of course, of different activities, different procedures. It has to be seen as a continuous process throughout the whole life cycle, and you need to maintain and manage the system as well. Last but not least, on the one hand, you need to stay always at the generally acknowledged state of the art with your activities, but also with your products. On the other hand, you have to follow a certain rule which says safety by design is always the first you should consider. Protection, second one. Information is the weakest one, and therefore the third option only. Not to forget about risks related to use error. We always have an interaction with users, with operators of those systems. So therefore you need to consider, okay, what is the status? What is their background? Do we have professional users? And of course, in almost all cases of in-house IVDs, we will see professional users. However, you need to consider the daily business of these people as well, and derive risks and manage risks as well. You need to manage all known and foreseeable risks, any undesirable effects, and you need to weigh them against the benefit at the end. The second chapter of the GSPRs, I do not want to dive too much into detail, but they come with very specific risks depending on the device characteristics. For example, contamination, EMC, electric shock, explosion, fire, also software, cybersecurity, and all of the individual aspects you typically can expect from different types of devices. Last but not least, not to forget about chapter 3 with the information, for example, residual risks. Typically, you also need to provide sufficient information on residual risks. As I already said, nothing comes without any risk. And this could be typically covered, for example, by putting them into the SOPs, setting up relevant trainings, etc. Okay. Let's switch to the standards. Standards, I already mentioned, one of the core standards, ISO 15189. This standard already comes up with certain requirements on risk management to establish a system. It's always pretty similar. So the idea and the mindset is always pretty similar: to set up a system, to assess the risks, to identify the risks, and also to reduce and manage the risks. What from my point of view makes it pretty specific here is that this standard especially talks about risks versus opportunities. So it also gives you the option to dive into opportunities. It's definitely important that you need to develop actions to both and to prevent any undesired impacts, whatever that might be, especially in the direction of patient care. This standard, as we can see already, is referencing 22367 as well. So it looks like that the combination seems to be a desired one already by the developers of those standards. One more time, This standard is also mentioned by name in the IVDR Article 55C, so therefore it is not officially harmonized by the European Commission, but it could be seen as de facto harmonized with a question mark because it's literally mentioned in the standard and therefore plays a major role here. In comparison, the next standard, ISO 14971, remember this is the one which is mainly focusing on devices and medical device manufacturers. However, again, the process described within that standard, again, is pretty similar to what we already see and what might be best practice: to have a plan, to have a report, to document everything, and to perform analysis, evaluations, reduce risks by controls, and do a proper risk-benefit analysis and let the system live again. This standard is more focusing on weighting risks versus the benefits. So the residual risks should not be outweighed by, or should be outweighed by the benefits at the end. It's important to understand that every device might have inherent risks, and you really need to dive into the potential problems which could come up with your setup, with your device, and also learn from while using the devices. The interesting thing here is that this standard definitely is harmonized with the IVDR, of course, again, for the focus of medical device developers and manufacturers. But it's a pretty good hint to have a look into the set annexes, because this annex directly connect the GSPRs with the chapters of this standard. So by applying a certain chapter of the standard, you get a certain conformity assumption regarding the connected GSPRs. So therefore, it's always worth to have a look into it as well. The other standard more related to the laboratory risk management is 22367 in the version 2020. So again, process pretty similar, but it is more focusing on the process itself. So instead of the devices, more on the processes, covering aspects from pre-examination, examination, and also the post-examination, but also including, at least partially, the design and development of laboratory examinations, which is pretty well matching the requirements of LDTs again. And as I already said, it's focusing on the handling and operation, but the concepts, the mindset, the technologies, techniques presented here are pretty similar to 14971. Last but not least, I would also like to spend one word on the new standard 5649 on laboratory developed tests. It also describes in some chapters, obviously, the risk management process, pretty similar, but with the focus on LDTs, again, for all of the stages of the procedure. But it also introduces a certain risk differentiation for the LDT, for the in-house device, which means that to put more emphasis, to put more focus on, for example, what is the importance of the information to the final diagnosis, or what is the impact of the result at the end. So really consider also these aspects, and also consider the expertise of operators, which typically can be found well established in LDTs. Okay, to sum it up, how does such a risk management cycle, according to state-of-the-art, look like? In the center, we have our own device, the in-house IVD. And first we need to establish and document a risk management plan for each device. This is what the IVDR wants to see. And this plan typically also contains the risk acceptance criteria. In a next step, then the cycle can begin. First, we need to identify and analyze all the known and foreseeable hazards by different techniques like brainstormings, etc. We should consider device risks and process risks as well within the workflows. We need to estimate and evaluate the risks. We need to eliminate, if possible, or at least control, which is reducing the risks. We call that risk mitigation. Again, consider device and process design. We need to individually reduce the risks, and we need to check and analyze the residual risks, the individual ones, but also the combined or overall one. And with that, we can do a risk-benefit analysis and demonstrate that the benefit outweighs the final residual risks at the end. During the usage of the device, after implementing it, we need to assess the experience, on the one hand, gained from clinical use, but also production, if production happens within a laboratory. If you really manufacture something within the laboratory, then you, of course, also need to ensure that the production is also considered in your assessments. Last but not least, if needed, and that is closing the cycle, you potentially need to amend. You need to learn from your doings, and you need to potentially amend control measures and update them if you find out that the original ones have not been sufficient. And all of these things need to be documented in the so-called risk management file, which more or less is a summary of a plan and a report and the individual records on the different findings. So my summary here is, first of all, IVDR has explicit requirements on risk management, which makes them binding for laboratories because it's an EU law. We know that standards are not mandatory. They are representing state of the art, but they are not laws. So it's always up to the implementers, the laboratories, to decide on which standards and if they use standards. But we see that it's highly recommended to consider standards here because they give you already a certain game plan to fulfill the GSPRs. We mentioned the 4 different standards here with different properties, and we need to establish and maintain a whole process for the risk management individually. And last but not least, we consider all of the aspects, on the one hand, on the individual devices, but also the process itself, so the steps in between. We need to manage everything, which means to reduce risks, to assess them, and finally to mitigate them. And last but not least, also to document that. And with that, I would like to hand over to Professor van Stapel, who will give us more insight into the daily business of a laboratory. Thank you.
Thank you. I will give you a short opinion of mine on how to implement lab-developed tests in the context of the IVDR, a practical way to go about it. As already mentioned by Matthias, the IVDR is here to stay. You will have to live with it. A very positive effect of the IVDR is that it will clean up the market of substandard devices. A negative aspect of it is that it actually creates quite a big administrative burden. If you compare it with the preceding IVD directive, there are many more administrative requirements and many more handovers between various authorities. So what I will try to tell is a way how can we bend the IVDR requirements to our advantage? How can we apply them wisely? The IVDR summarized very important aspect is what I call the intended use. And this intended use must realize a clinical benefit at an acceptable risk. And one of the first approaches to do that is a validation of your device. And the IVDR makes a distinction between what they call scientific validity. That means that in the literature you can find proof, or you have proof yourself of a quality that you can publish, that you have a performance achieved which was sufficient to demonstrate the clinical usability, the clinical benefit of your device. It's an important concept because it also means that with the analytical performance achieved in these studies, it actually proves that the clinical usability can be reached as well. In other words, it gives you a criterion to weigh your device on. And then the other thing is they ask you to validate the performance, and they make a distinction between analytical performance and clinical performance. In practice, this is usually done in one go. In order to validate your analytical performance, you have to validate it according to an acceptance criterion. And the acceptance criterion is, of course, clinical use. But anyhow, for demonstrating that to your competent authority, you will have to make this distinction in your files. You have to clearly identify these two aspects. And a last aspect of the IVDR is that the device must be safe. I distinguish here between 3 stages. The manufacturing, you have to guarantee the availability when the device is needed. The release of the device, you have to guarantee that it has a continuous acceptable performance. And then concerning the use, it has to be safe for the patients, relates to the clinical usability. Mostly, it has to be safe for the users, bystanders, and that relates then to all kind of effects like electrocution hazard or radiation hazard, things like that. Is usually not applicable, but it's actually what in the risk literature is called a precautionary approach. You try to avoid anything that is conceivable and hazard. And then at the end, you will have to make a statement, a summary statement that is the conclusion of what you studied here, and it has to prove that the benefit-risk ratio is positive. That is actually relatively hard to do. And then in absolute terms, one of the things that you can do there is anyhow try to argue that you reduce the risk as much as possible. So in the lab, there are 2 scenarios for the development of lab-developed tests for in-house use. The IVDR restricts them to in-house use. And the first item that we will discuss is a modified legacy device. And the second item that we will discuss is a de novo developed test. An in-house device, modified legacy device, you are allowed to introduce if the available devices on the market do not meet your needs, your clinical or operational needs, especially your clinical needs. Operational needs can have to do with also with benefit for the patient. How well is the whole organization making use of your results to the benefit of the patient? And if you don't find what you need, then you're allowed to make an LDT, provided that you can prove that the benefit-risk ratio indeed is better than what is available on the market. This requirement comparison with the market only will become absolute requirement when the IVDR is completely rolled out for the industry. So there are actually 2 scenarios in which you develop this modified legacy device. That is either you extend the intended use or you have an improved design. Now let's look at the risk-benefit ratio. On this axis, we plot the risk. On this axis, we plot the benefit. And the crosshairs is the original device that you want to improve. So if you extend the intended use, that means that the legacy device actually is has an increased clinical utility, and you are moving in this left quadrant where you are allowed to introduce it as an LDT. The other scenario is that you increase the reliability or the safety of the device. That's an improved design. And then you are moving in this allowed quadrant from the legacy device to the improved LDT. So you will need some kind of risk analysis to indicate why you did these modifications, and you will have to summarize that in the relevant documentation. For the in-house de novo developed tests, that means that you actually are developing a test that is not available on the market. And the general question of the IVDR is the benefit-risk ratio acceptable? And a first way to look at that is we always start from the intended use. We ask a number of questions. Which measurement are we measuring? In which sample? For which clinical diagnostic purpose? Is that the clinical diagnostic purpose that is listed in the IVDR? In which patients do we do this? The test is administered where and by who? And this you can translate in a failure mode effect analysis. You can translate the intended use in sources of risk and impact of failures. And the number of questions that you ask here is, first question is you compare it with the Annex 8 risk classification, because that is a classification which is used in the Article 5.5, and it also immediately relates to the impact of failures. Then you look at analytical aspects. Is the measurement well defined? Is it stable? Then you do an analytical performance validation, which, as I already mentioned, actually is linked to the clinical performance. Clinical performance is going to be your acceptance criterion. So does analytical performance guarantee clinical performance? And the last question, also an important question, is, are the different actors knowledgeable, competent? That, for instance, is important when you use devices outside of the lab, in a point of care test setting, or even directly by the patients. So let's now talk a little bit about risk evaluation and risk management. What I want to say here is that risk evaluation and risk management are not the same. As already mentioned, you have a number of hazards which combine into a risk determined by what kind of failure harm is going to occur, at which frequency, which damage is going to result from that. Can you actually do something about the damage? So a number of questions you're going to ask. The hazard, is that avoidable? The frequency of failure and frequency of harm, can you detect that timely to set up some kind of mitigation? Which damage will occur? Is it an acceptable, repairable damage? For instance, if your patient dies, that's irreparable and unacceptable. But there may be other circumstances where patient has to get just a second blood draw or something like that. That's much less of a problem. And then you have a number of tools mentioned in the standards about risk evaluation of risk management. You have the famous failure mode effect analysis, almost best linked to your validation file. You have you set up control procedures to detect results or an operation that is not according to your criteria. That will result in scrapping results, in rework of samples, producing new results. And then you look at the damage, at non-conformities. You have to have a system that captures these damages and non-conformities, and you have to identify sentinel events that are so serious that you will analyze them in detail. Now, risk management, that is trying to avoid harm and damage. So the best way of risk management is actually a failure-proof design that is designed for quality. If it can't break, then it won't break. Second best is what we call primary prevention, and that is best achieved by what we call process care. So the first thing that you do is you map the process, and then in that map process, you scrap what is not needed, what is superfluous. Everything that is there and is not needed can fail and give rise to problems, and you retain what is really needed. Then you harmonize your process as much as possible, because over all the processes that you are running in your lab, the higher the degree of harmonization, the easier it is to instruct and train your personnel, to cross-train them, to exchange them where needed. It's also a way to reuse what has proven its usefulness. It's also an important aspect to look at when you are looking at IVDR compliance. If you already did a validation plan for another device and you can reuse parts of that validation plan, then reuse them. Don't start doing the same thing over and over again. So very important in process care is that you organize communication up and down the hierarchy, and that you plan training and maintenance. And the third best is what we call secondary prevention. That is that you have a way to detect failures and to correct failures or to prevent damage. These are things like inline quality control, corrective and preventive actions, the management review, and it's actually a life cycle approach resulting in lifelong learning. In the end, you will have to draw up a summary conclusion file for your validation. And I make here a summary of what we just discussed. Risk analysis is the first aspect. You have a recipe for that could call that in the IVDR jargon, the risk management plan. At the design and validation time, the failure mode effect analysis is there one of the tools to use. And during the life cycle, you have to register all non-conformities, failures, and have a CAPA system. The outcome of this is reportable, auditable risks and CAPA inventories. Second thing is risk management, and the preferred way of doing that risk management is a process care primary prevention. The recipe is here to harmonize returning elements, reuse first-in-class validations, scrap what is not needed, failure-proof instead of inline controls, and plan maintenance. And the outcome of that is that you will have a lean process, which means that you have reduced production costs, and a clean process, which means that you have reduced failures and also reduced costs of failures. And the second approach in risk management is a life cycle secondary prevention. Recipe is here the inline controls, the registration of non-conformities, and the resulting corrective and preventive actions. And the outcome of that is cost of quality failures you will incur because you didn't really prevent them as secondary prevention. But it also will give you maybe a couple of preventive success stories to convince your management and your auditors that you are actually on the right track. And this concludes my presentation.
Thank you, Professor van Stapel, for the very interesting presentation. Thank you, Andreas Oberleitner, for your presentation as well. So with this, I would like to open the Q&A dialogue.
Thank you very much. Thank you very much, Florent, for the presentation and also giving such deep insights into the daily business. And I also like the point you emphasized, seeing it not only as a burden of the regulation, but also making an advantage out of it. So to see it really as a tool, risk management as a tool to improve your processes and to take everything which is possible out of it. We see that very often within the laboratories, and therefore my first question would be to you, from your point of view, what is the current status regarding the implementation of such risk management systems in the laboratories? So what do you think, how complex is the task? How far along are the laboratories? And what about the resources, knowledge, and employees? So is there sufficient knowledge, training, and also resources available?
As you already mentioned, risk management was an explicit requirement of the 15189 already in the 2012 version, and the 2022 version only makes that more explicit. That means that it also is a returning item for assessment by accreditation bodies. So all accredited laboratories, they have operating instructions on risk management. So your question actually translates to how effective is that risk management.
Mm-hmm.
With respect to the workload, if we now are going to apply this risk management to the topic of this webinar, the lab developed tests, up to now, most lab developed tests were actually not really presented for accreditation. The reason for that was that anyhow these tests have a low turnover and are they may be an economic burden on the lab because they require quite a bit of effort from the lab, but the return on investment is actually low, and the additional cost of an accreditation very often was avoided. That means that for these tests, maybe the implementation of the risk management system was not really up to date. In principle, the labs know how to do it because for their accredited tests, they were audited on this topic almost invariably, and they certainly know how to do it. The problem with the expansion to the LDTs now is whether they have the resources to do it. And in terms of the effectiveness of risk management in the lab, the auditors go and look for objective criteria which they can easily report on in their audit report. And very often they don't get much further than looking at whether there is a formal failure mode effect analysis which can be shown, whether they are done with a certain regularity. But whether the lab actually translates that into effective risk management, that's much harder to evaluate during an audit. If you want to evaluate that, you go to the management review and check how many success stories are available there.
Mm-hmm. Okay. What we have seen also in many laboratories already is when it comes to very specific cases, for example, generic groups or devices with a broad purpose. So how should an institution manage the identification and also evaluation of risk, especially for tests with a broad intended purpose as for exome testing, for example? And how would you suggest how should labs could best deal with risk management in general for a huge portfolio of similar tests, like generic groups, for example?
Yeah, as already mentioned in the slides, you always start from the intended use. Then you refer to large portfolios of similar tests. I also refer to reusing reusable elements in such large portfolios or in similar tests. So how do you define generic groups which could be give rise to such returning items? Can be similar measurements or pathogens or genomic sequences, etc., or it can also be similar technology.
Mm-hmm. Regarding practical examples for risk identification, you already mentioned some of them, or we were talking about some of them, but maybe can you again mention 2, 3, or 4 examples from your experience? What are typical risks a laboratory would see in their pre-analysis, in the analysis, even post-analysis process?
risks linked to the device that are typically risks that are linked to either the production of the device or the use in the analytical process of the device. So that is mostly linked to the analytical device itself or certain analytical characteristics of the device. For instance, the linearity of a test. And then I think of immunoassays and the famous Hook effect. It may be that if you have a very high concentration, that you give a false low concentration as a response. It has to do with analytical performance of the test. How do you recognize that? That's a risk at the level of the test. When the lab has analytical problems with the legacy device, it will usually tell the manufacturer about these defects or shortcomings. And we hope that with IVDR and the post-market surveillance, the companies will pick up these reports and will do something about them.
You already mentioned the manufacturers right now. When it comes to the analysis of device risks in in-house IVDs, they are often built on such components, of course, provided by the manufacturers. And for this purpose, they are somehow also dependent on information on the components, right? So this is particularly relevant for research use only components, but also for CE mark devices, of course. So from your point of view or the laboratory point of view, what would you need from the manufacturers, especially in terms of information on the components, to be able to set up your in-house IVD compliantly?
Yeah, when the manufacturer sells the product as a research use only, or as equipment for research labs or for non-medical testing labs, then for the manufacturer, there is no IVDR requirement. So the IVDR requirement shifts to the laboratory who is going to use these things. The IVDR limits such applications to in-house use. Again, for the lab, that is not too much of a problem because the lab normally doesn't sell these devices to other labs. They only use it for their patients or for patient samples referred to them, and there is no problem. However, the lab is required to make a validation file according to the IVDR and to communicate its findings with the competent authority and to be able to withstand a lifelong audit cycle by the competent authority. How this all is going to happen in practice, we have no experience yet. It will also depend on the Annex 8 classification. If it's a high-risk Annex 8 classification, then all countries are required to have this looked after by the competent authority. But some countries will actually expand the scope of what the competent authority is entitled to check to a much broader scope of tests. And the workload can actually become so big that both labs and competent authorities are going to drown in it. So I also don't know how this is going to be handled in practice. For lookalike generic applications, competent authorities will be confronted with an exercise in even-handed evaluation of the reports that they get from all the different labs, which are all required to, at an individual base, interact with the competent authority. So that's also a problem. One way out of that is that they introduce more guidance. The Medical Device Coordination Group at the European level is actually tasked with harmonizing how the competent authorities handle certain items. So that's one place where they can do it. The disadvantage of guidelines is that to the extent that the things are very can be generalized. You can use a guideline, but to the extent that there are very specific individual aspects, a guideline can be too rigid. The other disadvantage of guidelines and also actually of the IVDR and the detail of what they request is that if you switch to checklists that you have to check off, yes, I have it, yes, I have it, you risk that you never get to an effective management of your system.
Mm-hmm. I see. And is there anything which you would also think where the industry can help, so the manufacturers of components?
Yeah, I think the first way for the industry to help us is to make sure that we don't have to make modifications to legacy devices. So the more information, the broader they identify the intended use, and the broader they identify the type of samples and the additives and so on that can be used in samples, the broader these definitions, for them, the disadvantage is that they have to go through a more complicated validation file that they have to present with different authorities that they have to interact with. The advantage is, of course, that this is a one-time exercise that then doesn't have to be repeated by all their clients. So I think if they want to distinguish themselves from their competitors, they better do this. Also have to remind the industry that with the European tender regulation, such requirement can be actually an element of a tender on which they can be judged.
Mm-hmm.
So they have an advantage of actually working there.
Mm-hmm.
Then the other thing is there where it's for economic reasons and not practical or unfeasible to go for this approach, they could help us with anyhow for these exceptional cases to provide protocols. Protocols as such, they are not part of the IVDR. And protocols can be exchanged and used, and they anyhow would then stand as a reference document for the lab to fall back on, and for the competent authority also to fall back on.
Mm-hmm. Okay. Thank you very much for these insights. For the sake of time, I think I would switch over back to Simone.
Yeah, thank you very much for this very interesting discussion and dialogue. And with this, I would like to hand over to Andreas, who will give a demonstration to us on the PLATO-X IVD Assistant.
Yeah, thank you very much. I would like to give you a very small sneak preview on how the whole topic of risk management will be put into our platform. So I have already shared my screen. You should now see the platform again. So I'm logged in as a demo user, as a demo laboratory. And we see already a lot of green ticks here, because that is because we already went through the whole process with this demo project here. And as everyone who followed our past webinars already knows, that the tool itself guides you pretty well through the whole process, asks you for relevant information, and creates automatically, based on this information, your documentation at the end. So many things have been already pre-filled here. And if you scroll down, then we see already one more module. The new module here is the risk management module. And this module consists, again, according to our paradigm already in the past, of wizards collecting information from the user or on your in-house test, and documents which are created based on the given information. So for the sake of the presentation, I will just quickly jump into the sub-modules. So as we start, as we know, we need to start with a risk management plan. We have set up a so-called risk management plan wizard. I will jump into it. And as you can see, this wizard asks you about very basic, very fundamental questions, which are pretty essential for the whole setup of your risk management system. For example, we are asking if you have probably one or more of these relevant standards already implemented. This would then have an impact on the later filling of the documents. Then we are asking for where can we find your procedure. This is just for the sake of referencing and using it as a variable throughout all of the future documents as well. And we also need to define where the acceptance criteria are defined. So we know that risk management is nothing without responsibilities. So there is also a matrix, a responsibility matrix you need to fill out. And the tool helps you in remembering the 10 typically important responsibilities you need to assign within the organization. This is already pre-filled by some dummy names, as you can see here. And then we also need to balance risk versus the benefit. And this is what we are doing here. We need you to write up the final device benefits at the end, which is also reused in the documentation. Last but not least, you need to set up your FMEA scheme, which means to define what are your criteria, first of all, for the severity, but also for the probability. And then you need to define which of those facts are acceptable and which are not. I think you're pretty familiar with this concept of the risk matrix and the assignment of probability and also severity. And based on these informations, you're already done with the simple risk management plan wizard. And what it does, it pre-populates. I will just quickly zoom in to increase the size to enhance the readability. Again, it pre-populates you the template we provide on the risk management plan. You see all of the variables are already available, like the standard applied, also the relevant references to this standard, the product information, which is known already from the former wizards from this demo project. The responsibility matrix is depicted here again. Changes will be updated immediately if you change it in the wizard. And also the description of the acceptability criteria. Of course, you can alter this document as you like and also export it to your own QM system. But just with a few clicks and filling in some information, the whole risk management plan is already set up pretty fast. The typical next step normally in risk management is that you need to start thinking about the risks. And for this, we are implementing a so-called risk analysis studio. In this demo, only 3 risks are mentioned here. You can easily add risks, and the whole system works a little bit like a Kanban board. So the idea is to bring risks from left to right through the process while identifying, assessing, and mitigating them to make the risk level finally acceptable. Very briefly, to add a risk, you get a new screen where you need to define hazards, the harm, and also the severity of harm, because that typically cannot be altered easily, while the probability is something you can work on. And you also typically need to describe the risk event leading to these hazardous situations at the end. And by clicking through this process, I will probably, I have prepared a very small example. Sorry, it's the wrong one. Where I will quickly fill out that with some dummy text. So for example, my hazard would be, for example, an inappropriate sample storage. In my example I prepared here, which leads to the problem, the potential harm that a patient issue could not be detected, and therefore the patient would not be treated. And this is pretty high, so it could lead already to a permanent impairment or irreversible injury, right? So the risk event leading to such a situation also needs to be filled out. And for that example, we could say For example, what if this sample storage is left not in the controlled cabinet where it should be? With that, I can create this risk, and the risk appears as kind of a ticket or item or element here. And by clicking on it, I can now start working on it. So first of all, I need to add the probability. We know that, I think, pretty well also from the typical process of an FMEA. And I say, okay, the probability is the second lowest one in that case, and I save it. And then immediately it shows me, okay, with that first assessment, I need to do something. Control measures are needed because the risk, according to my original setup, to my original risk acceptance criteria, is too high. So by clicking on it, I can now be guided through a process. I need to place a control measure here. This is what you typically need to identify and define here. And I also need to demonstrate it. This is needed because without demonstration, of course, you're missing the link, but this is in many cases probably very simple. So to say, okay, in my case, it's probably an SOP I want to link here. Let's say it's a sample a storage SOP or something like that. Let's call it ST-SOP1 on storage, maybe, with some release dates, first 2023, and typically it also has a version number like 2.1. And with that, the document is added, and we have now our evidence deposited here. So the good thing is here, the yellow label disappeared because the control measure is there, and I can have a final look onto it. Okay, I'm good with the evidence document, I'm good with my defined control measure, and now I'm able to save it. With saving it, I need to finally do the residual probability check, and normally the probability should have been reduced by introducing my control measures. The original was 2, as we remember, and now we say, okay, we reduced it from 2 to 1, and finally I can do the assessment of the risk. The assessment is done automatically, and the tool tells me, okay, everything is good. The risk level with these settings is now acceptable. We are good to go to the final column. Yeah, and this is how we can manage the risks individually in a very smart and easy way. And by going back, then we see that a new file appeared, which is the risk management record. I just want to scroll quickly over it because it sums up all of the individual risks fully automatically. You do not need to copy paste anything. Everything is in the file, and for the case of presentation to your competent authority, you can easily put up this document. Last but not least, we also need to create a report on that. For that, again, there is a simple wizard, because before we finalize the report, we typically also need to be sure that, first of all, our defined criteria have been met, that everything is completed. We also should put an evidence here. I will just write another SOP here, and here is another document. We say everything has been completed also in the review process. Again, I need to document it in another document. And last but not least, to check, do you consider all estimated values for the probability? So those are the questions you need to ask yourself in the process. And here you can document your decisions. Let's say everything is done. Save and create. And finally, we see that based on the given information, again, template on the risk management report is available and is fully filled with all of the given information, with all of the variables fully automatically. Yeah, you just need to finalize these documents and are good to go to continue in the process. Yeah. That's what I wanted to show. As already mentioned, the tool will be released soon. We are continuously working on this software product and improving it. So your feedback is always highly appreciated, of course. And with that, I would like to hand over back to Simone.
Thank you. With this, I would like to thank the guest speakers for their contribution and, of course, the whole Platomics team, especially to our software development team. Thank you for your attendance. Stay tuned for our final ABD Ready webinar in June, and I wish you a pleasant afternoon.